And we are back with another, what is this called? Science session. Okay, we're gonna talk about modulation of rates of transcription. Okay. Where are my notes? We're here. So transcriptional regulation will control the levels of gene expression by modulating the number of RNA polymerase II molecules transcribing a gene. So if you have one gene, you have gene X, okay? There's one gene X, but if you have one RNA polymerase II, you get like one result. But if you have 10 RNA polymerase II, then you can get a lot more genes, a lot more proteins. Okay? So again, when only one RNA polymerase II is bound to the gene X, the transcription level would be low, and because you would only get, minimal mRNA production. But if you have like 10 RNA polymerase II molecules bound to gene X, then that's when transcription level would be high, and that would lead to a significant increase in mRNA production. And that regulation will ensure that the genes are expressed at appropriate levels depending on the cell's needs and environmental signals. So you have a picture that is obviously not shown there. You have multiple RNA polymerases on a single gene, and the dark spots would represent the RNA polymerases, and the continuous regions between them would be the transcribed gene. And again, that is for proper regulation. So there's different rates. Different genes can have varying levels of transcription. So for example, you have one long DNA strand, you have gene A and gene B on it, because again, a gene is just a specific region on the DNA. So gene A, its transcription might be faster. It's transcribed at a faster rate, and thus you have a greater amount of RNA being produced there. And this RNA is then translated into a larger quantity of protein, and that's represented by multiple protein A's. On the other hand, you have gene B, which might have a slower rate of transcription, which would then result in fewer RNA, and thus because of that, fewer RNA can be translated to protein B, and you only have a few protein B. And the next is the lac operon in bacteria. It is a model system for understanding gene regulation. All right. Where is the... Let me just check if the character thingies... Yep, it works. And then if I try, what's up? Welcome back to another science session, definitely trial. For the lac operon in bacteria, it is a model system for understanding gene regulation. And with that, it has different components. You have lac Z, lac Y, and lac A, and the promoter and the operator, and the regulator gene would produce a repressor gene. So this repressor gene would normally bind to the operator region, which would prevent RNA polymerase from transcribing it. So, however, if you have lactose, the sugar acts as an inducer. The sugar would bind to the repressor protein, and that would prevent it from binding to the operator up there, and thus the RNA polymerase would then be able to transcribe the lac operon genes, and that would result in the production of mRNA, and is thus translated into the enzymes to break down lactose. And this is a very, very good example, because if you have That's because if you have too much lactose, you would need to like break it down, right? And that's why lactose itself could serve as a regulatory signal, because again, it would bind to the repressor changes in conformation, it can't bind to the operator, and thus the RNA polymerase can then transcribe the thing that would destroy or just break down the lactose. And then again, in eukaryotic cells, the majority of protein regulation occurs at the transcription level. Okay. Majority of it would occur at the transcription level. In what way? That means that the cell primarily controls how much of a particular protein is produced by regulating the process of transcription, aka the DNA into RNA. And in about 25,000 human genes, around 2,000 encodes proteins. Again, in 25K human genes, around 2,000 encode proteins involved in transcriptional regulation. And these regulatory proteins would control when and where and how much of a gene is transcribed into RNA, and thus it would influence protein production. And this control would then allow cells to respond to various levels or signals and can maintain the gene expression level. And we mentioned it before, but there's a very, very good picture here about the different TF2 stuff. So you have the activators which enhance transcription by interacting with the enhancer region, and that it could be located far from the transcription start site. And these activators recruit the general transcription factors such as TF2A, B, D, RNA pol 2. Meanwhile, repressors bind to silencers. So again, activators, you have activators and repressors. Okay? It would bind, those are proteins, and those would bind to specific gene regions, aka enhancers and silencers. Nice. And then you have the Tata box, and then TF2D, you have the regulatory regions. That, again, I mentioned it. Those are enhancers and silencers. The 5' flanking region and the promoter regions are key for transcription initiation because they play a critical role in the binding of transcription factors and RNA polymerase. KK? Nice. Beta globin gene family expression is regulated by a distal locus control region. So the beta globin gene family is regulated by the distal locus control region, which is upstream of a cluster of stuff. And this region would play a role in that's a specific one for the globin genes. We have the enhancers and silencers, those are on the genes. It is around 30 to 15 nucleotides upstream from the start site. Hey, I mean, the transcription start site is 32. Yeah, it's the promoters. Upstream activating sequence acts similarly to an enhancer. And we mentioned the rest. Spacer DNA also. And then you have the mediator complex. Yeah, okay. Let's go through this one. So you have the bending of DNA and mediator complex. Alright, so in eukaryotic transcription, gene regulatory proteins would play a crucial role in gene expression by interacting with regulatory sequences in the gene control region. And these sequences, which can be quite distant from the actual gene X would include promoters, the tata, and spacer DNA. And the interaction of these proteins with the transcription factors in RNA pol 2 would be a bridge to connect the transcription machinery with the regulatory regions. And that's why this process would then result in the synthesis of RNA transcript. You then have the mediator complex mentioned earlier. What is the mediator complex? That is a multi-subunit protein complex that will facilitate transcriptional regulation by bridging. What does it bridge? It bridges the gene regulatory proteins and RNA pol 2 machinery, and it's present in both the yeast, the S. cerevisiae, and humans. Although their specific subunits differ. The S. cerevisiae would be med 1, 2, 6, 7. And in humans, it's also med 1, 2, 6, 7, but it has the CDK molecule. What is that for? The CDK molecule would be important for cyclin-dependent kinases, CDK8 and 19, and that would help it with gene expression as well. Those are important, I think, for... What was it? It was for the cell cycle. We can talk about that later. Yes, and then you have the GAL4 protein in yeast, which would have two main functional domains. The GAL4 DNA binding protein, which would bind to the tata box of the gene of for galactokinase. So again, GAL4 DNA binding, and then you have the GAL4 activation domain also, which would be for the transcription of it. And then you just have different transcription factors. That's alright. Next. Alright. So we have a full flow here. In transcription, let's see. In transcription, gene regulatory proteins such as activators and repressors would bind to the DNA sequences in the enhancers or silencer regions near the gene. Good, we talked about that. And these regulatory proteins would then recruit transcription factors, yes, which would have DNA binding and activation domains. Okay. Once the transcription factors are bounded to the promoter, DNA binding domains help the factors bind to the promoter region, yes, and the promoter region contains the tata box, yes. Once the transcription factors are on the promoter, that would then attract the basal transcription machinery, which is the tata binding protein. And the others, mediator complex, would consist of multiple subunits, and it plays a vital role by linking the gene regulatory proteins to the machinery. Okay. Sounds like a balita. Sa ulo ng mga nagbabagang balita. Anyway. We talked about these. Next, you have DNA binding structures, which would include several function motifs. Okay. We talked about the zinc fingers, which would use zinc to stabilize the protein loop. And then you have the basic zipper, the leucine zipper, which is for dimerization. So the zinc finger again would be for protein loops. Basic zipper for leucine would be for dimerization. And then you have the basic helix structure, which would be for binding and dimerization. And then the helix turn, that's fine. Okay. Around 2,000 known transcription factors are involved in regulation of enhancers and silencers. Yeah, and those are important for gene regulation. Varying amount of molecular mechanisms are known. And yes, those would be complex interactions. There's a specific gene right now. It's for the liver. That's the transthyretin, I think. Transthyretin gene, which is liver specific, and that features key regulatory regions for its expression. The enhancer region is located approximately 1.96 kb from the gene, and that would play a vital role in gene activation. There is the repression by Wilms tumor protein. And no, my paper. Let me just arrange it. And that is just the EGR control region. WT1 binds to a specific site within the EGR1 control region, and that would lead to the repression of the gene. There's also the kidney tumors, okay, in early development, and the inheritance of both maternal and paternal genes for the Wilms tumor genes can lead to a situation where expression of the gene is reduced or eliminated. So if you lose the Wilms tumor protein repressor function, if it can't repress anymore, this could affect the expression of other genes and lead to kidney tumors. So it's really important in early development that you keep your WT repressor function. Okay? But yeah. And in this example, the expression of well, you have a lot of different examples. You have 1, 2, and 3. Expression number 1, there is competitive binding between the activator and repressor. So the activator has a binding site on the DNA, but the repressor also competes for the same one. Wow, it's like harem. No, that's a love triangle. Oh my gosh. Okay. The vision. So you have the binding site for for the repressor, and also the binding site for the activator. It's like, and it overlaps. And thus, the repressor can inhibit the function of the activator and prevent the transcription machinery from happening. And that's a form of repression. Next, you have the masking, okay? Masking of the activation surface. So they could have different regions, but the surface of the, what was it? The repressor binds to a region of the DNA overlapping the activator's activation surface, and that would lead to the activator being prevented from properly interacting with the transcription machinery. So both can bind to different binding sites, but then even if it can bind, the top part of the activator the part that should interact with the machinery is prevented. That's SAG. Or just SAG. And you have the third example of transcription repression, and the mechanism involves direct interaction between the repressor and the actual transcription machinery. So in the picture, the repressor is a little bit nearer to the TF2D, which has the TBP, and then it would just stop it from working. It's like tapping its shoulder and like, no bro, don't transcribe. And then the TF2D with the TBP on the Tata is like, okay bro, I won't transcribe. Funny. Next page. 3 molecules. You have cortisol, retinoic acid, and thyroxine, which would again play a role in transcription regulation. Cortisol is a steroid hormone, and it is produced by the adrenal glands. It binds to the glucocorticoid receptors, which can then act as transcription factors to regulate genes. You have the retinoic acid, which is derived from vitamin A. And then you have thyroxine, a thyroid hormone. So again, cortisol, made by adrenal glands, a steroid hormone. You have retinoic acid, made by vitamin A, thyroxine, thyroid hormone. All three could turn things off or on. Then you have the variable region, the zinc finger region, mentioned before. So you have different response elements, okay? Okay. You have the glucocorticoid response element. It is recognized by what else? The glucocorticoid receptors. Estrogen response element, vitamin D response element, thyroid response element, retinoic acid response element. So those have different response elements, and the arrows of it would also go in different areas. So glucocorticoid would be N3. Well, it's like an arrow from the left to right, right to left. It's GRE, ERE, VDRE, and etc. Right? And then now, let's talk about the glucocorticoid hormone activation versus their thyroid hormone activation. Okay. So, when the glucocorticoid, upon entering the cell, let's talk about the glucocorticoid first. Upon entering the cell, it would just go through the plasma membrane and bind to its glucocorticoid receptor. Once it does that, take note, the GR or the glucocorticoid receptor is initially attached to the HSP90, the heat shock protein 90. But if the glucocorticoid hormone attaches to the glucocorticoid receptor, the receptor changes form. It will then be released from, it would then be released. So it's not attached anymore to the HSP90. At the bottom of it, the NLS, the nuclear localization signal, is then revealed, and it would allow the complex to enter the nucleus. Once inside the nucleus, it would bind to the glucocorticoid response element, aka on the gene. Once that happens, it can facilitate transcription initiation with the assistance of RNA polymerase II. So again, glucocorticoid attaches to the glucocorticoid, what's the R? Receptor. Glucocorticoid receptor. And the glucocorticoid receptor, which was initially attached to the HSP90, is now released, and the nuclear localization signal is now seen. It now has a flag, and it can go through the nucleus, where it binds to the glucocorticoid response element, and then it can do transcription. But then for thyroid hormone, it's different because it passes through the plasma membrane either by diffusion or a carrier-mediated transporter. So it can do diffusion or through a transporter. It will just go through straight until the nucleus. And once in the cytoplasm, the thyroid hormone binds to the thyroid hormone receptors. And the hormone receptor complex may either bind to the DNA before or after it dimerizes. Dimerizing is just partnering up. The thyroid hormone would bind to the TR, thyroid receptor, and the thyroid receptor could either go to the to the thyroid regulatory element and then dimerize, or dimerize before binding there. And dimerization typically would involve a RXR, retinoid X receptor. And that is essential for the complex's ability to bind to the hormone response element on the DNA. And this binding results in the activation of transcription, with the RNA pol 2 initiating the gene regulation. this page is crucial. Yeah, different DNA binding domains. Wait, my notebook. Do not fall apart. Do not fall apart. Okay, it's alive. Okay, the glucocorticoid action to the GR homodimer mechanism involves different steps. It would enter the cell, binds to its receptor, the glucocorticoid receptor in the cytosol. The GR has 3 domains: activation domain, DNA binding domain, and the ligand binding domain. And in its inactive form, it is bound to an inhibitory protein, but once the hormone binds to the ligand binding domain at the top, it will, we mentioned this, it will undergo a conformational change, exposing the signaled nuclear localization signal. It then dimerizes to form a homodimer, and then it would translocate to the nucleus, binds to GRE. Nice. And you have T3 and T4. Wow. No T1. Okay, so the action of T3 triiodothyronine in the cells begins with T3 and T4 being transported from the bloodstream across the cell membrane, and once inside the cell, the T4 becomes T3, and the T3 is a more active form, and which then goes to the nucleus. So T4 from the bloodstream crosses the plasma membrane. T4 becomes T3 using 5' deiodinase, and then once it is in T3, it will go to inside the nucleus. It binds to specific DNA associated thyroid hormone receptors, and that would belong to a larger family of hormone receptors. Good. Let's just... There's so much sticky notes. No. Okay. Thyroid. Let me just note this. Thyroid. I need a bigger sticky note. Found one. Oh no. My folder is blue, but my sticky note is also blue. So I can't see it. Has to be blue, like blue. Okay, thyroid hormone elements consists of 2 repeats of the hexameric half site. It is a conserved sequence that enables the thyroid hormone receptors to bind and regulate gene expression, and the orientation of these half sites can vary. Yes, genes are... There are different genes positively regulated by T3, the growth hormone, glucose transporter, and etc. Okay, let's talk about the absence or presence of T3. So this is a good one. I really have to note this part down. Okay, there. So, if you don't have T3, the thyroid hormone receptor and the RXR would... These are the first. They do not activate gene expression. So again, if you don't have T3, the thyroid receptor and the RXR will not activate gene expression. Instead, they will recruit co-repressors such as NCOR, SMRT, HDA. What are they? So again, just to make sure it's instilled. If you don't have T3, the TR and RXR will not activate gene expression. It will instead recruit 3 things. NCOR, HDA, and SMERT. NCOR would be nuclear receptor co-expressor. Nuclear receptor co-expressor. And then you have SMERT, silencing mediator for retinoic acid and thyroid receptors. Wow, that's a mouthful. Again, NCOR is nuclear receptor co-expressor. NCOR. And then SMERT is silencing mediator. Silencing mediator for retinoic acid and thyroid hormone receptors. What is silencing mediator? SMRT for retinoic acid, SMR, and thyroid hormone receptor T. Silencing mediator for retinoic acid and thyroid hormone receptors. And those pairs of co-repressors would bind to HDA. What is it? Histone deacetylase. Histone deacetylases remove acetyl groups from histone. If you remove acetyl groups, that would cause the chromatin to bind really tight. It would become tightly packed. That would be chromatin condensation. That would lead to it becoming more inaccessible. Okay? And if it is inaccessible, the RNA pol 2 will not be able to bind. It is inactive. Inactive. Yeah. So, once more, another word. No T3. That means the thyroid receptor and the RXR will not lead to gene expression. It will instead call 3 of its inhibitory friends, the NCOR, SMRT, and HDAC. And nuclear something co-repressor, nuclear receptor co-repressor, silencing mediator for retinoic acid and thyroid hormone receptor, and histone, and those two will call HDAC, histone deacetylase. Histone deacetylase will remove the acetyls from the DNA, from histones, I mean, which would then lead to the chromosomes being more tightly packed, and then it's inaccessible. The RNA pol 2 cannot attach, therefore no gene expression. But then, if you have T3, now we're moving to a different realm. The T3 is present. It would form a TRT3 because the T3 binds to the thyroid receptor, and then it would dimerize with the RXR to form a heterodimer. It would then call more friends, the TRE. I mean, it would bind to the TRE, which contains like a sequence. And then what friends will it call? It would call Drip Trap, SRC with hat. Drip Trap, who is it? It's vitamin D, receptor interacting protein. Vitamin D, receptor interacting protein. D, R, I, P, vitamin D, receptor interacting protein. Or Drip Trap, the thyroid hormone receptor associated protein. Those are the same things, but Drip Trap sounds cooler. Vitamin D, receptor interacting protein. What's SRC? Well, the SRC would be steroid receptor co-activators, which would bring in HAT, histone acetyltransferase, I mean. The HAT would acetylate histones, which would then decondense the chromatin, and the DNA is now more accessible. RNA pol 2 can come in and boom, gene expression. So, if you have T3 binds to the TR, you have TR3, dimerizes with RXR, calls Drip Trap, the vitamin D receptor interacting protein, or the thyroid receptor associated protein, which would then call it the SRC as well, the steroid receptor co activator, which would have or call in the histone acetyltransferase, which will acetylate the histones and will decondense the chromatin. RNA pol 3 can come in. Good. Let's note T3 with T3. So and this is just another part regarding the histone deacetyltransferases. Okay. So in eukaryotic cells, normally DNA is wrapped around histone proteins, which would form, or which is known as a histone octamer. And the histone octamer, the interaction is critical for regulating gene expression. Okay, so normally the DNA is negatively charged. It will bind tightly to the positively charged lysine residues on the histone. So if you deacetylate the histones, that would the strong positive charge on the histone would be removed, and then the DNA who was negative, who was looking for a positive partner, would be like, where are you? So it would then, wait, one more time. Yeah, the positive charge on the histones would keep the DNA tightly bound if the histones are deacetylated. However, when they're acetylated, the acetylation would neutralize the positive charge, and this weakens the interaction. So again, if you deacetylate it, the strong positive charge on the histones would keep the DNA would keep it bound, tightly bound. But then if you acetylate it, it would neutralize the positive charge of the histone. And then it can now, and it will be like, wait, I can't see it anymore. So it will just loosen up. Okay, car. Market, I'm a market. Diagram illustrates the feedback regulation system. Yeah. This part would be that this would involve the hypothalamus, anterior pituitary, and thyroid gland. So we start with the hypothalamus. It would release the TRH. Hypothalamus releases TRH, the thyrotropin releasing hormone, which would then, and the TRH, if the anterior pituitary sees that, oh, there's TRH, it would then release TSH, the thyroid stimulating hormone. And then, once that happens, the thyroid gland is like, oh, there's a lot of TSH, I will make thyroid hormones. So again, we start with hypothalamus makes TRH. If there's a lot of that, anterior pituitary is like, I'm gonna make TSH. If there's a lot of that, thyroid gland is like, I'm gonna make thyroid hormones. And then the thyroid hormones would then that would be negative feedback on the anterior pituitary and the hypothalamus above. Because, it would, if not, then it will just keep on producing thyroid hormones. Duh. So that's why it's good. Alright, now we have JAK-STAT. Okay. Interferons play an important role in the immune response. And that would activate transcription via the JAK-STAT system. And that's triggered by the binding of interferon ligands to the respective receptors on the cell surface. Okay, so first, let's start properly. Just in case this becomes confusing. Let's not confuse it. You have interferon A. You start with A, okay? Interferon A. That would the receptor the interferon A would bind to the receptor, and that would lead to the activation of that JAK1 and type 2 kinases. The JAK1 would phosphorylate themselves and the receptor. So interferon A binds to the receptor. That would lead to phosphorylation of the JAK1 and type 2. And that would activate the STAT proteins. It's a curvy protein. Phosphorylated STAT proteins would form dimers together. And those would then go inside the nucleus. Let's start again. Interferon A binds to the receptor. Receptor would phosphorylate itself and the JAK1 and the type 2, and phosphorylation of those would lead to phosphorylation of the STAT. Phosphorylation of the STATs would then lead to it dimerizing. Dimer form of the STATs would then go inside the nucleus, and then the P48 protein is involved in that because it would bind to the interferon A response element along with the STAT dimer, and that would then boom transcription. But then if you have interferon A was alpha, and then now if you have interferon gamma, you have the JAK1 and the JAK2 kinases. Interferon gamma binds to the receptor. JAK1 and JAK2 would be activated, and it would phosphorylate the STATs. And then, it's kind of similar. After it dimerizes, it would go inside the nucleus and binds to the gas, the gamma activation site. So, yeah. JAK and STAT. The JAK-STAT pathway. And then next is you have a signaling network involving a lot of things. We can talk about this later, but it would be fun to just try it a bit. The signal pathways would be initiated by growth factors or stress, most likely stress, and that could trigger different receptor-mediated processes at the cell surface. And the growth factors typically activate receptor tyrosine kinases, and while stress factors may trigger a range of other signaling molecules, such as like FAC and RAC, you still have others such as the MAP kinase pathways, and that consists of different kinases: the MAPK, the MEK, ERK, and etc. We'll talk about that in a next video maybe. And then you have you have calcium and phospholipase C. So we start with, this is a big picture. Wait, let's breathe. Okay, let's breathe. So it would start when receptors on the cells, the surface, such as immunoreceptors or receptor tyrosine kinases or GPCRs are activated. Those would stimulate phospholipase C, gamma and beta, which would break down PIP2 into IP3 and DAG. IP3 and DAG would lead to release of calcium ions from the ER because IP3 through the IP3 receptors, while calcium can also influx into the cytosol using release activated calcium channels. Calcium release activated calcium, CRAC. More calcium will lead to binding to CAM, and then you activate it. And then inside you have NFAT as well, nuclear factor, etc. There's a lot of things going on. And then the last part of this one would be metallothionein. And that is a protein involved in metal ion binding. And that's specifically zinc and copper. and those would have cysteine residues on it. And metallothioneins play critical roles in detoxifying metals, regulating metal homeostasis, and that would also protect the cells from metal toxicity. And the binding of metal ions to metallothionein would help store and shuttle metals in and out of the cells, particularly under the conditions of metal stress or deficiency, and this would also allow the protein. So yeah, to, gene regulate or apoptosize or modulate. And then regarding how you want to regulate the transcription of it, you begin the process with a steroid receptor protein binding to the GRE, an enhancer region in the hormones. It's in the steroid hormones that would interact with specific proteins. Then you have the transcription factors. And the next, the metal response element MRE would act as the regulatory site, which binds to the MRE activator protein. And that's where the Tata box comes in. You also have the regulation of it. Again, the cytosol, the metal response element MTF1 factor 1 is present in an inactive state. When zinc and cadmium enters the cell, it binds to MTF1, causes of change. MTF1 goes inside the nucleus, and note, H2O2, not water. It can also induce the activation of MTF1, but the mechanism might be different. So once inside the nucleus, MTF binds to MRE and would initiate transcription. Voila. Those are very specific terms, but we'll get through them. Okay. Yes. What's up? Yeah, we are DNAing the DNA. It's going through the dinosaur ages. Welcome back, definitely Lendor and George. Shout out. Okay, actually, I would like to go and discuss DNA repair and recombination also. Would that be fine? That's fine. I will. Let's go. Okay, DNA repair and recombination. So DNA damage, you have single base changes and double-stranded breaks. Okay? And those are the two major classes. But there are other things that could be like other specific stuff. For single base, that type of DNA damage refers to the alterations where only a single base is changed. Point mutations, or you have replicative misincorporation. And that would, well, for point mutation, that could be a silent mutation or missense or nonsense. Silent mutation, you don't have a change in the protein. But missense is there is a change in the amino acid. There's also the nonsense mutation where, Sabrina Carpenter's song. You also have the nonsense mutation, which would be like the premature stop codon. Okay, so we finished DNA transcription, and now we have DNA repair and recombination. Alright, so finished with a single base. Next is double-stranded breaks. These are more severe because you broke both. A more severe form of DNA damage where both strands of the DNA helix are broken. And the double-stranded breaks are very detrimental and could lead to chromosome fragmentation, translocations, or loss of the genetic material if it is not properly repaired. Double-stranded breaks are usually repaired by either NHEDG or HR. NHEDG would be non-homologous end joining. Non-homologous end joining. HR is homologous recombination. So we can just talk about that later. Wee. Okay, DNA bases are susceptible to 3 types of chemical damage, I mean. How? In what way? You have hydrolysis, deamination, and oxidation, and methylation. So how my notebook, oh my gosh, you can hear pages. That's my notebook just falling apart. Okay, there you go. Maybe there's too many pages. It's fine. Okay, for hydrolysis and deamination, cytosine can undergo deamination. That would convert it into uracil. And this could be bad as uracil pairs with adenine instead of guanine. Adenine can be deaminated into hypoxanthine, and that could lead to mutations. It's just overall bad if you deaminate it, okay? And then oxidation, guanine is more highly prone to be oxidized. That's also, and thymine and cytosine also, which could then can interfere with base pairing and could lead to replication errors. Methylation, you have cytosine can be methylated to form 5-methylcytosine. It is a modification that can interfere with base pairing. It's just bad, bro. So just don't. But it happens randomly. Sad. Very, very sad. Okay. And hydrolysis, and dimerization of adjacent pyrimidine. Oh, note. UV light can cause DNA damage to DNA by inducing dimerization of adjacent pyrimidines. You know what? I would like to drink water in a bit. Okay, I'll do that in a second. I want to get through DNA repair first. Yeah, of course. So no, we were talking about UV light can cause damage to the DNA by dimerizing the pyrimidines. What are the pyrimidines? So if purines are adenine and guanine, pyrimidines are cytosine and thymine. So if you dimerize the pyrimidines, that's bad. Because the dimers could distort the DNA structure and block proper DNA replication, which would lead to mutations. And the presence of these dimers is a form of DNA damage that, if left uncorrected, it can cause skin cancer and other DNA-related diseases. Yes. And DNA repair mechanisms such as nucleotide excision repair are responsible for removing those. So if you dimerize the pyrimidines, that is awful because it can wreck the DNA structure and could lead to mutations. So that's why the nucleotide excision repair is responsible for removing the dimer, and it could then, restore it to its proper form. Hooray. And carcinogens like benzoapyrene, BP, found in tobacco can also react with DNA to form base adducts. Don't smoke. And that is bad because those are covalent attachments of carcinogen to the DNA molecule. And this process begins when the BP undergoes oxidation by CP450 enzymes. And that would form an epoxide intermediate. The intermediate is highly reactive. It can bind to DNA. And if not repaired, it can cause mutations in tumor genes such as the P53 and RAS. And that's all. And that's just because of tobacco, bro. So don't smoke. First-hand or second-hand smoking, sad. Please don't. An accumulation of such mutations can lead to cancer development, and the formation of the adapts and the mutations are very, very awful because they can cause they cause essential. There are essential mechanisms through which the carcinogens would exert their genotoxic effects. Okay. So which of the following spontaneous lesions in DNA occur frequently? It is depurination. And it is the loss of a purine base. It is a process which happens when the bond between the sugar and purine base breaks. A lot of them. There's also what are the products of deamination of cytosine and 5-methyl. So cytosine, uracil, C, cytosine, and 5-methyl cytosine. That would be uracil and thymine. C, where's the structure of a cytosine? Yeah. Oh yeah, if you remove the methyl of it, it becomes a thymine. Because cytosine is, it's like this. You draw piatos. Piatos first, okay? And then, what was it? NH2. And double bond O. And then N, or that's cytosine. But thymine would be close enough. Oh, it's deaminating, right? Deaminate. Yeah, if you remove the amine, it becomes a thymine. Full coat. And then if you remove, no, I mean, if you deaminate cytosine, it becomes a uracil, but if you deaminate the 5-methyl cytosine, it becomes thymine. So it becomes uracil and thymine. You have 4 + 1 principal repair pathways for single bases. That's the base excision repair, which is, you know what? Actually, we can save this part for the next one. No, we can do it. Base excision repair would be, okay, first, you have a deaminated cytosine, okay? The deaminated cytosine became what? Became a uracil. Cu became a C, became a uracil. It is recognized by the uracil DNA's glycosylase. And then, the uracil DNA glycosylase would recognize it. Would cleave the bond between the uracil base and the sugar phosphate backbone. And then now you have a missing base over there. The AP endonuclease would see, oh, it's missing a thing. It would cut the sugar phosphate backbone, and there is now a gap in the phosphate backbone. So again, the beginning would be, you have a deaminated C. This is first pathway, okay? This is base excision repair. The first part is, oh, that's why I'm hungry. I see. Yeah, bros, I'm gonna have to eat. That's why there is a lag time. Okay, I think that's it for that topic for now. Bye bye.